Okay. Good afternoon, everybody. We are really sorry not to be able to be today with you, but we are in fieldwork, actually using a lot of the methodologies that we are going to explain today on our presentation. I'm Sara Garcês, and this is my colleague Hugo Gomes, and today we're going to make a presentation about prehistoric art methods and analysis and classification. And actually, we're gonna do a very, a presentation that actually will explain some of the projects that we are working currently and that we are using the methodology that we're gonna speak about it. And some of the results that we are actually also having in the last years, and a lot of them you also can find them published already. We're going to give you some tips for bibliography that can actually help you for this presentation. So today I'm going to start with explaining that, generally speaking, and most of you will know, rock art can be found in many different types of sites. The two most obvious, and probably it's the most where we can actually find most of the rock art, is either in caves, like this one. This is Maltravieso cave in Spain, but we also can find rock art in open air sites. And in both sites, we can find both engravings and paintings, okay? So it's actually quite current to have both techniques in many different places. Both engravings and paintings can be found open air or in caves. But many times, for example, when we find engravings near rivers, it's actually only the engravings. We don't have normally painted sites near rivers. There is some examples like in Koa, but it's quite rare to find paintings near rivers. What we can find mostly in Iberian Peninsula, for example, it's caves with prehistoric art. Could be very old or more recent rock art. And then we have a lot of these type of sites, open air shelters with schematic paintings. This is normally the case of Portugal and Spain. And this is the kind of rock art that we can find, a painted panel, okay? It's very important for you guys to know the difference between paintings and engravings. Or inside caves, we also can find schematic art, but normally it's in these kind of sites that we find more Paleolithic art, due probably to conservation issues regarding paintings, but this is also the kind of rock art that we can find inside caves. But actually, no matter if we are talking about caves or open air sites, working in analysis of pigments or dating rock art or any kind of archaeometric analysis, actually it's we can call it a discipline. It's an area of rock art research that actually involves a lot of different disciplines and knowledge. So it's not only taking into account that we are talking about archaeological sites, but we have to work in prehistoric art questions. We have to know about photography. We have to know and have geologists in our team. This is a very important aspect. How sites actually exist and what and the shape of the sites, how do I actually impact on the rock art conservation. We have to be aware of geography characteristics. We have to know about topography, biology, obviously chemistry and physics. So it means that actually doing an archaeometric analysis or work in a rock art site involves a lot of different disciplines, and all of them are very important. It's not that we have to know more about archaeology than other things. The sampling methodology also has rules or guidelines, actually. Obviously, we have to first have permission to be on the sites, and it's very important that permission is given to do the sampling work that it's supposed to do. But when we go to a site, we have to take into account different aspects of the panels, of the rocky support, of the figures itself, And the most important thing is always the conservation purpose of the site. So we don't take samples just because. We have to have very serious scientific questions to do this work, and then always be aware of the conservation issue regarding the site. So normally a collection of sampling or a collection of samples has to be aware of the typological spectra of motifs. So what type of rock art are we dealing with? If there is a variation in the pigment, if there are superimpositions on the figures. So what actually we have to first question of what do we want to know about the site, that only archaeometric analysis are possible to actually help us to understand the sites. There are some ethical extraction techniques that we have to be aware also, and we always use guidelines. Normally, we go through the American Institute of Conservation guidelines. There are a set of guidelines that help us to extract samples in a very careful way, with very specific rules, just because for the conservation of the site. There are preferred sites in the panel that we should extract pigment. We have to consider the fractures of the panel, if there are painted parts of the figure. There are specific instruments that we have to use, like sterile tungsten scalp and tubes. And obviously the choice of where we extract the sample is very important, also for conservation issues regarding the site. The scientific questions have to be associated with the archaeological context, obviously, the geographical area, the coloration that we are working with, if it's red, black, white, yellow, orange, or other colorations, the overlapping of images, because this actually can help us a lot with the chronology, the different techniques that are applied to do this rock art, the typology of figures, and if we know or if we can work this in advance, the chronology of the figures. So what would be our questions, our scientific questions regarding the site? Why do we want to take samples? All these issues, all these parts of the research have to be in mind and already helping us to answer of our scientific questions. Okay, now I'm going to move to Hugo that will explain about the methodologies.
So welcome to this archaeometric class that I will give. I will not be further, of course, in this physical and chemistry approach, but just explain you which kind of methodology and protocol we developed in the last 15 years, studying rock art, archaeological evidences, mainly associated with rock art. And as Sara explained, we have different contexts. So we have different chronology of pigments. We have different kind of pigments. We don't know. We start these from a very huge base that the pigments are made with natural raw materials. And we start to develop with the projects and after also in my PhD, a kind of protocol of methodologies to achieve, in fact, all these questions that Sara mentions in the slide, in the last slide. So very briefly, we use the methodology that we have nearby our institutions in IPT, in the Ferrara University, and also with other collaborations that we start to develop in this process of developing this protocol. We start off obviously using the optical and the stereoscopic microscopy to analyze, to see. It's the first approach to the sample that we catch. Of course, now we already develop also the protocol, and we start to use, for example, portable apparatus, portable mechanisms that for some of this information, we can start to collect them in the fieldwork that help us, for example, to choose the best sample to collect, to bring to the labs. Then we have the Raman spectroscopy that is very common to the study of pigments in general, not just in particular the prehistoric ones. The X-ray microfluorescence, the scanning electron spectroscopy, and in particular, when we start to think in the receipts that could be made by prehistoric people, we join, for example, the gas chromatography and the Fourier, the FTIR analysis, because we start to identify some of the organic part that usually with the Raman, with the XRF or with SEM are not so easy to identify. So here is just an example of a microstratigraphy that we have the lucky to found. We found a piece that is considered a little bit bigger than the samples that we usually collect, and we have the opportunity to make this slice in the sample. And as you can see, we have the entire sequence of the panel. We have the substrate, the blue gray area. Then we have a line of natural concretions that, of course, the earth is a living planet, so everything is constantly erosion, creating new crusts in the surfaces of the rocks. Then we have here a human evidence that are a part of these two figures that in fact is represented by this red line. And in the top of this, we were also recognize a new concretion line that grows after the painting was made. And the black dots that you saw in the first approach, we think that could be it's a charcoal, pieces of charcoal that could be mixed in the pigment receipt. But when we start to look in particular in this zoom, we catch that it was an accretion of a recent fire, and in fact this small charcoal became stuck in the wall after the paintings. So Raman spectroscopy is very useful for identify the chemical mineralogical composition of these pigments. We can sometimes identify also the production process and the subsequent alterations that and reactions that happen in the pigments. We are talking about mainly iron oxides, for example, and we know the sequence of the alteration of the oxides. If there's water, if there's lack of water, the minerals change in natural way. And of course, with this, we can also identify some degradation process of the walls and also from the paintings itself. XRF is a non-destructive method, so we keep the sample for other methods. For example, this is one that could be also used as a portable apparatus to go in the field, to go in the shelters, in the caves, to make a general analysis of the walls. The problem is sometimes when we have calcite layers over the pigments, of course, the results, it will be just calcium related with this, and we don't achieve the real composition of the pigment. And the other problem is that it is not useful for identify, for example, the organic material that could be preserved in the pigments. With the SEM, the scanning electron microscopy, we are able to see, for example, the processes of preparation. We can see, for example, the grinding process. If you have kind of fibers inside the pigment, we are very, with the scanning of the sample, we are able to recognize this kind of structures. And of course, with the analytical, the ADS analysis, we can identify the kind of material that is made. Using the gas chromatography, of course, this is a kind of consequence. We used first general approach to identify the materials, and then if we have a kind of a suspicious that we have organic binder, organic compound inside of the pigment, we use gas chromatography to recognize the type, if it's a vegetable, if it's a animal glue, something that could be mixed with or inorganic part. The FTIR is very practical because it gives us very quickly and very clean the elemental composition of the sample, increase the sensibility of the analysis, so we can search for very particular peaks. For example, we can identify which peaks are part of the substrate rock and which peaks are part of the pigment itself. So, and is also very useful because we can also detect some organic substance. So we can also apply gas chromatography in the sequence of FTIR. It's also very important to resume that we can apply one method, but in general, it's very important to use a multi-proxy approach, using different methods in the same sample to achieve, because all the methods have limits, all the methods have particular aspects. So when we start to use XRF, Raman, FTIR, gas chromatography, and others, we complement the information that we get from these small samples that we got from rock art, usually paintings. This slide is salient to push up the importance of the identification of binders that usually is very hard, very difficult. And of course, we think that is related with the conservation issues of organics, the degradation of the walls that usually cover and don't help us to recognize very easy the organic part. And of course, we are talking about paintings that are old, so this organic part could be already completely disappeared in the receipts that we can imagine that were used. So this is a research that's still ongoing. Of course, we have, as Sara tell, we have a lot of publications related with this in the last few years. Some of examples that we made in this is one rock shelter in Bacarense de la Serena in Spain, that in fact we have this organic identification with FTIR, and it was very useful then to continue and to start to organize our protocol, taking into account that we have organics. We could not identify which kind of specific organic material was used, but with further studies, we probably can identify if it was a plant, it was a cactus or another organic material. Also, very interesting was the recognizing of some paintings made with charcoal, that in fact we try to make absolute dating with charcoal 14, but the quantity was really tiny, and it was not possible to achieve a good and a scientific result. This is another example. Then you can search all these papers in ResearchGate and Academia.edu. They are free for download. Another example that we developed, it was with a collaboration with a master student, in fact, that works in Brazil in Serra da Capivara, and we were also able to recognize the organic component that in this case we identified that were made from plants. Then we need to think that the paintings in Brazil are much more recent than the Paleolithic ones and the schematic ones that we are dealing here in Iberia. So again, the conservation and the utilization of the organics in this case was fine. This is the publication that is also free. if you don't know rock art about Serra da Capivara, go to see and search, because it's an amazing world of rock art. And then we continue and we extend this small protocol to a huge project, the First Art project. And I will pass again to the word to Sara.
Explain very fast that all these methodologies that you just learned about, we are actually using it in a project called First Art. First Art deals a lot with uranium-thorium dating. It's a very recent debate about how old can rock art be in the world, and some exercises, especially in the Iberian Peninsula and Indonesia, have been done using uranium-thorium to actually try to date the calcite that is under and on the top of pigments. And in First Art, we actually are trying to use both documentation techniques and methodologies with archaeometric analysis to try a more comprehensive and a better understanding about the rock art that we are working with. Also using absolute dating with the uranium-thorium and C14 methods. And we are also trying to work in this new research line that is trying to determine if we can find presence of human DNA, old DNA, also in pigment sampling. So First Art is actually trying to put together a protocol, not only of archaeometric analysis, but also with documentation and dating of rock art. And I think very soon we will have some results, some interesting results about this project. So in generally speaking, and resuming the presentation, what a multi-proxy analysis can be possible to do with a mixture of different archaeometrical methods in one rock art site. We actually can characterize pigment components. We can actually identify raw materials, production processes, so how we will explain the way that possible recipes of pigments could be made. The application techniques is also very important because it can or not define the chronology, and then we can actually mix with also absolute dating and other comparative methodologies. And in the end, with a broader knowledge of a rock art site, we actually can apply conservation measures. And this is a very important aspect of conservating the site, but taking into account the chemical characteristics of the site, the physical characteristics of the site, the rock art itself, the archaeological context. So everything must be also used to apply conservation measures in a rock art site. So this is our presentation. Again, sorry that we are not there, but actually Professor Pierluigi can answer some questions that you may have. Don't hesitate to contact us if you have any doubt, and have a rest of a good course. Thank you.
Thank you.